So this will be the first course on bioinformatics. The instruction is you can see the activities symbol at the bottom of the screen. Then followed by that you can click on polls option. Then you can click vote. So for those who have joined, please try to answer the questions that are posted on Google Meet polls. So you can click on activities tab, then polls, then vote. If someone is facing any problem in accessing, please unmute yourself and I hope everyone can access the polls. So coming about this session. So in this session, I'll be taking a little summary part for the last 5 week lectures. Followed by that, I'll be taking up some sample problems which are related to the assignment as well as the week lectures. Then I'll be taking questions from portal as well as questions from students. So in this session, I would also like to demonstrate on how to use EMBOSS tool. So this will be after the sample problem session. There will be a demo on how to use the EMBOSS tool. So first, to start with our understanding on what is bioinformatics. So in the field of biology, we define bioinformatics as the combination of biology and different technology aspects, where most of the cases we use computers as our trusted allies in unraveling the mysteries of the living organism. For example, can we imagine a world where the complex details of biological data are seamlessly organized, managed, or analyzed with the help of advanced computational techniques and information technology? So, this somewhat is precisely known as bioinformatics. So, what areas does the bioinformatics include? Bioinformatics is known to be a highly interdisciplinary field that draws upon various domains.
How to mute myself?
Yeah, sorry for the disturbance. So, coming to that, it includes various domains including biology, medicine, mathematics, physics, and computer science. In bioinformatics, biology is the fundamental science providing the raw data and the context for understanding living organisms at the molecular level, cellular, and organism levels. This biological knowledge is important for the development of computational methods and algorithms for the analysis and interpretation of biological data. When it comes to medicine, it also has a crucial role in bioinformatics by integrating genetics and molecular information into clinical research and practice. Bioinformatics in medicine is widely known concept, which involves applications such as personalized medicines, pharmacogenomics, and analysis of disease-related genetic variations. Mathematics provides a theoretical foundation. That is, mathematical models are employed for tasks such as sequence alignment, structural prediction, statistical analysis. I understand these are the examples that you are going to come up in the future week courses. Physics helps in understanding the structural aspects of biological molecules, particularly in structural bioinformatics. Then comes the important one, which is computer science. It is termed as a core of bioinformatics. So, which helps by providing the tools, techniques for data storage. So, once we store the data, we can analyze them, or whenever we want, we can collect or we can see the data. So, the algorithms are software applications which computer science relates are developed to process and interpret the biological data efficiently. So, to summarize, I can tell bioinformatics acts as a nexus or connections, leveraging insights from biology, harnessing computational power of computers, utilizing mathematical models, and incorporating principles from physics and medicine to advance our understanding. There are some major aspects or aims of bioinformatics to consider. The first one is a well organized database. So before starting up, can anyone let me know what is the term database mean, or any example?
Organizing a particular data, organizing in the sense curating and annotating.
Okay, that's good.
And
Okay,
some examples are NCBI, PubMed, PDBJ, etc.
yeah. So can I tell our mobile phone is a database? So how many of you accept? Can raise your hands. Yeah, I hope a couple of hands raised. Yeah. So in our mobile phones, right from organizing our contacts to our photos or gallery, the best example is a gallery, how it organizes the media from our camera, media from our downloads, media from our documents. So in bioinformatics, the role of databases is to create and maintain biological information such as DNA sequences, they are long, protein structures and etc. So these databases in bioinformatics helps in efficient data retrieval and analysis, enabling researchers to explore and understand various aspects of biological systems. Next comes hypothesis. So this we make hypotheses to identify some patterns or relationships or potential correlations within the biological data, which lead to hypotheses that can be tested experimentally and contribute to a deeper understanding of a biological process. Next is a web server. So web servers are user-friendly tools, can we tell, or platforms that enable researchers to access and utilize computational tools and resources remotely. Next comes the concept of virtual screening. This is widely used in drug discovery. So computationally, using this, we can computationally assess the potential binding of molecules. So which helps in identification of promising candidates for further experimental validation. So these concepts are one. I would consider the fifth one, which is the big data analysis, as the one, because being a bioinformatics people or being from biological background, the problem comes in handling the challenges that comes with the vast amounts of data. So these limitations could be overcome by using bioinformatics tools. So here, okay, today for answering these questions, we'll be using the chat box where I'll see answers from the chat. So I would like to start with a basic question, which tells which interdisciplinary aspect does bioinformatics leverage to advance our understanding of living organisms? Yeah, you can use a chat box to answer this. Okay, those are joined late. So welcome to this session once again. So we are starting with the summary to the week 1 lectures. So I'll be taking sample questions as well as summary simultaneously. Now this is the question people can answer. And when you have any doubts, please feel to ask at the same moment. And there will be separate dot solving session at the end as well. Yeah, I can see a couple of answers as T. Yeah, all of the above. As I mentioned, bioinformatics is an interdisciplinary field which uses various domains including biology, medicine, mathematics, physics, and computer science. So the answer is all of the above. Next, I have one more. What is the primary role of computer science in bioinformatics? We have discussed computer science as one of the interlinked field with the bioinformatics. So what could be its role or support from computer science we get in bioinformatics? I hope everyone can see my screen. Anyone facing the problem in viewing the screen or in my voice audibility? So here, the computer science helps in the computational tools or techniques which will be useful for data storage, retrieval, and analysis. Next, I hope I have one more. So I have discussed about virtual screening before. So what could be its objective of virtual screening in bioinformatics? The more details about virtual screening and other things we'll be discussing in further week lectures. This is just to acclimatize you to this problem solving session. These are questions are arranged in such a way. Okay, I see some answers B coming up. Yeah, virtual screening is a technique where we try to predict the interactions between biological molecules. So generally, what will be the biological molecules? Proteins, DNA, RNA, and some other small molecules. Mostly, this technique is employed to predict the interactions between proteins and small molecules. Like for example, given 100 small molecules, you can easily find the predict the interactions between these 100 molecules with the protein given. Yeah, next to go. So the central dogma of molecule is a framework. Yeah, thanks for your answer. Anu. That describe the flow of genetic information within a biological system, particularly in cells. So it was first proposed by Francis Crick in 1957. So this has three main processes to briefly talk about. It is replication, transcription, and translation. So coming to replication process, so this we have genetic material usually in the form of DNA, and it is duplicated to create an identical copy. So the replication is crucial for transmission of genetic information from one generation to other generation. Next is the transcription process. So in transcription, a segment of DNA is used as a template to synthesize a complementary RNA molecule. So this molecule, generally mRNA we call, which carries genetic code from DNA to cellular machinery responsible for protein synthesis. Next is the translation. So during this process, the information encoded in mRNA is converted into a protein. Okay, so subdisciplines within the bioinformatics. It being interdisciplinary, bioinformatics is itself a multidisciplinary as well. So it has various subdisciplinary things which deals with the biological data analysis, interpretation, and application. So first thing is genomics, which deals with the study of entire set of genes within an organism, or a genome, study of genome. Next comes with the proteomics, which similarly involves the study of whole set of proteins. Followed by that, we have structural bioinformatics, which helps in analyzing and predicting the three-dimensional structures of biological macromolecules such as proteins and nucleic acids. Followed by that, we have functional genomics, which understand the role and functions of genes, proteins, and along with their interactions. Other subdisciplines include phylogenetic analysis, systems biology studies, metagenomics, pharmacogenomics, and many more. Okay, here I have one more question, which tells in which process of the central dogma does the synthesis of functional protein occurs? Yeah, thanks for your answers. Yeah, as I mentioned, this is the last step in central dogma, that is translation, which helps to translate the information contained in the RNA to the protein. Yeah, in a couple of minutes, we'll see how this mRNA information in mRNA is converted to protein. Yeah, then I have one more. We have discussed genomics as one of the subdiscipline. So what could be the primary focus on genomics in bioinformatics? The genomic annotation is nothing but then it's a naming criteria. We'll get to know when we read in structural biology, structure related features. Sorry. Yeah. Yeah. Here, the primary focus of genomics and bioinformatics is exploring the entire set of genes within an organism. So with this, I'll start with the first concept in bioinformatics, that is sequence analysis. So sequence analysis is fundamental. So because it involves the examination and interpretation of biological sequences, as these sequences carry genetic information which is essential for understanding various biological processes. To start with the first, DNA. Here I told sequence analysis, but I have given sequence structure. This is just for understanding. But here our primary goal is for understand the DNA sequence or order of DNA, how they made, composition of DNA, to be brief. So each DNA sequence to us is unique to an individual and is inherited from one generation to the next. DNA is composed of 4 nucleotide bases: adenine, thymine, cytosine, and guanine. The sequence of these bases along the DNA molecule is what constitutes a DNA sequence. That is, the arrangement of these 4 is termed as a DNA sequence. In a DNA sequence, there is a concept called complementary base pairing. In complementary base pairing, A pairs with T and C pairs with G. So this complementary base pairing ensures the stability and double helical structure of a DNA. So what does a bioinformatic do in reading a DNA or understanding a DNA structure? So we used to get meaningful information from DNA sequences, which we can aim to understand functions of genes and non-coding elements within the genome. Once DNA is done, next comes the protein. So proteins are essential macromolecules in living organisms, performing diverse functions such as enzyme catalysis, structural support, signaling, and transport. Here, protein sequence information, like protein sequence study or analysis or information, refers to the specific order of amino acids in a protein molecule. It's just like order of your roll number 1, 2. So this first place, this amino acid, second place, this amino acid. Yeah, that. So the sequence of amino acids in a protein determines its function, structure, and also interaction with the other biological molecules. So proteins are composed of 20 different amino acids, each with unique chemical properties. The sequence of amino acids in a protein is dictated by the sequence of nucleotide bases in the corresponding gene. So the primary structure, we have different levels of structure in a protein structure organization. So the first one is a primary structure, which refers to the linear sequence of amino acids. That is, order of these amino acids. These amino acids are linked by a peptide bond, as shown in the image below. So here I have one question. So how many different amino acids are present in proteins? Yeah. Thank you for your answer. So we have 20 standard amino acids that are present in proteins. Next, in a DNA, what is the complementary base pairing that ensures the stability of double helical structure or the formation of that structure? Or in simple, you can tell what is the complementary base pairing observed in DNA. Yeah. Observe the options carefully and answer this question. There is a twist in this question. Yeah, so here the complementary base pairing happens between A and T as well as G and C. Yeah. Next, so we'll start with the decoding. So we have mentioned in a process of translation, the information in mRNA is decoded to form a protein. So this information from gene is read out to build a protein. So we have a concept called codon table. Codons are nothing but a group of 3 nucleotides. For example, when we take a alphabetics ABC, ABC can be a codon. DEF can be a codon if ABCDDF are type of nucleotides. So this codon table, what I am showing here, see, codon table is nothing but 3-letter combinations of nucleotides. So this codon table serves as a universal guide for translation of genetic information into the protein. And there is one concept here. When we see, there is a degeneracy. Degeneracy is nothing but different combinations of these nucleotides can lead to similar type of amino acid. So for example, if you see the glycine is coded by 4 different ones. So this is known as a codon degeneracy. And when we observe in this thing, we have stop signals as well as a start signal. AUG, which is universal start codon, which encodes for methionine, initiates the protein sequence or initiates the protein synthesis. And we have UAA, UAG and UGA has stop codons, signaling the termination of protein synthesis. Next, now we know these 3-letter triplet nucleotides can form, but how these are read in a frame? So then the concept comes reading frame. So this reading frame determines how mRNA sequences is divided into codons. So on in other terms, we could tell is a specific way of grouping the nucleotides into sets of 3. So there are 3 possible reading frames for each strand of DNA. As we know, DNA is a double-stranded, so each strand of a DNA has 3 reading frames, frame 1, frame 2 and frame 3. So here, for example, consider the DNA sequence given ATG, TCG, GAA and TAA. So what are the 3 possible reading frames would be is that the first reading frame, which starts from the first nucleotide, that is ATG is 1 codon, TCG is 2nd codon, GAA is the 3rd one and TTA is the 4th one. Next, coming to the frame 2, it starts with the 2nd nucleotide, that is now we are removing this A and starting from T. So T, G, T. Then the second one is C, G, G. And the third one is A, A, T. And finally we left with the 2, combination of 2. Next is the third one. It starts with the 3rd nucleotide. So removing first 2, A and T, we are directly considering 3rd one, G, G, T, C. This way. Further, these 3-letter codons are converted into amino acids using the codon table. So here, before moving on to something, I have some questions related to permutations and combinations. It's a mathematical kind of questions. Yeah, if you can answer, please answer. I'll be explaining the solution. These are nothing to do with bioinformatics, just understanding for our next concept. Yeah, first answer the top one. How many 2-letter words can be formed using letters A, B, C, D, allowing the repeats? can see some different answers like okay, B. Just for now, answer to the top one. We'll come down to the 3-digit number one later. Yeah, thank you, Swapnil, for the answer. Yeah, we'll do. Yeah, okay. I'll start with the explanation of the first one so that it would be easy to solve. Allowing repeats means that these letters can include AA. Yeah, so a two-lettered word. So first, let's comes to the first letter. How many ways we can fill the first letter? The first letter can be filled by 4 ways. That is either by A, either by B, C or D. The next letter, since we are allowing repeats, the A or the first A, B, C and D can be used again. So the second place as well can be filled with the 4 times. Okay, just give me a second. Let me open the Jamboard. Consider we have a box here like this. So in the first position, we can fill this by 4 options. That is either by A, either by B, either by C, either by D. Similarly, in the second one, we can fill this by 4 options. So overall, we'll get 16 options to fill. So in the first case, we have 16 ways a two-letter word can be formed using letters A, B, C, D. So let's take the next one. Yeah, so don't go to the formula of permutations and combinations. Yeah, here the answer is 27 because 3 different places and 3 options to fill in each place. So we get 27 ways of filling. Yeah, as I explained in previous, we have again 3 spaces to fill to form a 3-digit number. So first place could be filled either by 1, 2 or 3. So we have 3 ways to fill the first one. Similarly, the second one we have 3 ways, and similarly in the third digit we have 3 ways to fill. As mentioned by Aniruddha also, the formula is 2 to the power n for 2 digit and 3 to the power n for 3 digit. That's a permutation combination formula. So totally we have 27 different digits can be formed. I hope it's clear how 27. This is just the mathematical practice. So next we have one question related to codon. So how many codons, code for stop codon in the genetic code? Can anyone tell what is the role or what does a stop codon do? I hope I explained it. Transcription, terminate synthesis. Yeah, that's correct. It will terminate the protein synthesis whenever the protein translation program encounters this. There is a termination, and as many has mentioned, the answer is 3. We have 3 stop codons, 3 codons that code for stop codon. Next, we'll start with the one of the sequence-based feature. So the first one is a nucleotide composition. So this nucleotide composition refers to the distribution of ATGC nucleotides in a DNA or AUGC nucleotides in an RNA sequence. That is percentage of these. The next one is GC content. So GC content refers to the total number or percentage of G or C nucleotides in a DNA molecule. Similarly, AT content. Just like I have shown a formula of GC or GC content, GC ratio here. So we count number of G's plus number of C's divided by total length of DNA molecule, or number of A's, number of T's plus number of G's plus number of C's into 100 gives the GC ratio or a GC content. Similarly, AT. In above formula, if we replace in numerator GC with A and T, it gives the AT content. Next, finally, we have amino acid composition. Amino acid composition is distribution of 20 amino acids in a protein. Okay, to start, I have some of these questions. Try to answer this. So if AT content in a DNA sequence is 28%, what is the percentage of GC in the same DNA sequence? So to answer this, see, yeah. So the overall percentage we term it to 100%. So when the AT content itself takes 28%, the remaining percentage is for GC. So 100 minus 28 gives the GC content. So 100 minus 28 is 72. This is a basic question. Next one. Oh, sorry. Yeah, okay. Before moving to the next question, I would like to introduce a concept of char graph rule. So in the 1950s, a biochemist named Charvin has discovered that amounts of nitrogenous bases were not found in equal quantities in a DNA. So okay, just give me a second. Let me open some. Yeah, just give me. So in chart graph we have two things, first parity rule and second parity rule. So I'll come to these rules after answering this question. So if the T content in a DNA sequence is 12, so what is the percentage of AT and GC in the same DNA sequence? Yeah, it is 24. So before moving on to this, let's go back to the this char graph rule again. Yeah, I have little problem with internet connection. I'll just give it. Yeah, so first thing we have a rule of basic composition. So in a DNA molecule, the number of adenine bases is equal to the number of thymine bases, and number of guanine bases is equal to the number of cytosine bases. That is A equals to 2, A equals to T, and G equals to C. And we have a rule of base pairing. That is, adenine always pairs with the thymine through two hydrogen bonds, and guanine always pairs cytosine through 3 hydrogen bonds. So according to this here, so since thymine content is 12, we expect adenine to be also 12. So someone wants any doubt? Please keep your mics muted so that it won't create disturbance to other students. Yeah, so here we have answer is D, that is 24% and 76%. AT is 24, T is 12 and A is 12, and G we have remaining, that is 76. Next we have a concept of biological databases. So initially we have read what are the aims of bioinformatics. So the first one is to develop a well organized databases. So currently we have a wide range of databases available to store and manage biological data. So these databases cover diverse aspects of biological information, including genetic sequences, protein structures, functional annotations, and more. So can we check some of them? Broadly, they are like structure databases, sequence databases, and others. First one to discuss is PDB. So generally contains three-dimensional structure of biological macromolecules, primarily proteins and nucleic acids. Next we have nucleic acid database. Information on experimentally determined nucleic acid structures in DNA and RNA. Then we have some sequence databases, GenPank, which is a comprehensive database of nucleotide sequences. Then we have UniProt, which is a comprehensive resource for protein sequence and function information. And other databases we include functional and pathway related databases, Gene Expression Omnibus, Interpro, which gives us other annotation related information. So with this kind of summary is done, now we will be taking up some of the sample problems. So the first one to start with is average bonding energy of the sequence ATAGC is. Yeah, I have seen different answers here again. Okay, let's go to the Jamboard. So the given sequence we have is A, T, A, G, C. Right? So let's get the dinucleotides from this. So A, T, T, A, G. Everyone can see my jam screen, right? White screen where I'm writing. Yeah, thank you for confirming. And finally we have GC. And what are the corresponding energy values given? So here for AT is -7 and TA is -7, AG is -9 and it is -12. Now let us take the sum. The sum will be -7, -7, -14, -14, 9, -23, -23, 12 will be -35 divided by number of pairs possible, 4. So the answer is Yeah, here I see someone asked what is energy mentioned in kilocal or kilojoules. So that there are different ways. In an average, the hydrogen bond energy is taken to 1 to 5 kilocal per mole. When we go to joules, it is 25, and when we convert it to kilocals, it up to 6. So these energy values are just given for this solving purpose. Just like we take the equilibriums in force fields and all. Okay, here is it clear or anyone needs an explanation again? Sorry, previously my jam was stuck. Now I solved the issue. This is clear, right? Thank you. Please be open to talk. Whether you are right or wrong, you shouldn't be here, but you shouldn't be wrong in the exam. So that is the whole purpose of this live problem solving sessions. Okay. Next, okay. We'll move forward. I think the answer is A. Okay, next thing is number of trinucleotide units obtained from a DNA sequence of length 20 nucleotides. Just now I explained the concept of overlapping dinucleotides, where in the ATGC, in the previous problem, we have extracted the dinucleotides. So how many overlapping trinucleotides I can obtain if I have a DNA sequence having 20 nucleotides? Okay. I'll wait for one more minute. If anyone has any other answer other than okay, further no more answers, I'll go forward. Yeah, we have 18. So if we have a length of 20 DNA sequences, a DNA sequence of length of 20 nucleotides, we can get 18. How? Okay, let's move on to Jamboard again. Yeah. So I'll take a shorter one just for explanations. A, T, G, C. Now let's try to extract the possible trinucleotides, overlapping ones. So the first trinucleotide is what? Can anyone tell? Unmute or in the chat, please. Yes, that's correct. ATG. One is fine. Yeah, ATG. Now let's move on to overlapping. Next one, T. The next letter is T. Make a trinucleotide possible with this TGC. Next, can anyone tell what is next after TGC? Yes, that's correct. Further, we have only GC, so that we can't form a trinucleotide. Okay, now we have done this. Okay, let's move. Let's take other one. Little bigger. ATCCGG. So the possible trinucleotides will be ATC, TCC, CCG, CGG. So for a sequence of 6 nucleotides, we could only extract 4. So here the formula is n minus 2, where n is the number of nucleotides here. Similarly, let's go back and check in this. So we have 4 nucleotides. So n minus 2 is 4 minus 2, which is 2. So we got the 2. So here we have a length of 20. That is, n value is 20, n minus 2 is 18. So 18 is the answer. Next, I have given a DNA sequence which is CG, TTT, GAC. So how many total hydrogen bonds would be there between a base pairing? So consider the given sequence is one strand sequence. So how many possible hydrogen bonds could be made to the next strand, complementary strand? I have some answers. 20 hydrogen bonds. Okay, let's move to the Jamboard before moving on to solution. So here it is the given sequence. So to its complementary strand, whatever it is, how many bonds that C make? Yeah, 3. So 3 hydrogen bonds we have for here. We have 3. How many will G make? Same, 3. How many will T make? 2. So similar again, T, 2, 3, 2 plus 3. So the overall sum will lead to 6, 10, 15, 20. 20 hydrogen bonds are possible. Okay, is this to answer 20 is correct? Fine for everyone? Okay. So this is how the DNA base pairing will happen. A and T will form 2 hydrogen bonds and GC will form 3 hydrogen bonds. Given we have studied the concept of complementary base pairing. So what would be the complementary strand for the given DNA sequence? Remember, the directionality of the DNA is also important. Yeah, no need to type the whole complementary sequence. You can select the options, which will be the easier one for you. Yeah, before going on to the solution, I have Is there anyone who is attending their first problem solving session? First one. Please, you can raise your hand. Okay. So this is your first. You previously have not attended any sessions. Okay. I understand many of you. I'll be a little slow here. Yeah, please down your hands. I got the count. Yeah, thank you. Yeah. Okay, let's move on to Jamboard again. So this is the given. Okay, sorry for the delay. So now with that we have 3 dash here in the start. So the complementary and start and stop. So let's find the sequence first. The complementary base pairing of A will happen with T. T will happen with A. G with C. G with C. C with T. Is it correct? Yeah. So here it's not correct. So C should be with G. T again with A. A. C and T. And the double stranded of DNA will run in opposite directions. So now this is 3 dash and 5 dash. So the answer is B here. Next, how many nucleotides are needed to code for a protein that is 100 amino acid long? Yeah, every one of you can answer this because these are not graded, just for your practice purpose. Yeah, this is according to our codon. So triplet, 3 nucleotides combine to form 1 amino acid. So in order to form 100 amino acid long, we need 300 nucleotides. Okay, so this is the codon table. So the combination of 3 nucleotides gives to 1 amino acid. Okay, next it's a true or false question. Is it easy to break a bond between A and T than G and C? Yeah, it is true because we have only 2 hydrogen bonds between A and T, and we have 3 hydrogen bonds between C and G and C here. Here is the next one. So which among the following in EMBOSS is used to find the complementary strand for a sequence? Yeah, that's correct, Chevi Bandari. Thank you for explanation. Yeah. So it's reverse sequence. I'll demo how to use EMBOSS online version, and if time permits, I'll also demonstrate. Yeah, I'll also demonstrate download, how to download and use it on your local system. Just there is couple of questions more left. So what is DDBJ stands for? Yeah, I'll explain the available options in EMBOSS along with the installation process, how to install it. Yeah, so DNA data bank of Java is DDBJ. Next, which of the following amino acids has only a single codon? For this, you can refer to your codon table, which is available in the online. Yeah, tryptophan is coded by only one, and other one is methionine, which is a start codon. So GenBank contains the information on for most of these concepts, the explanation is not given here because it's already discussed in the week classes. Yeah, GenBank contains information on nucleic acid sequences. And there is some special information about GenBank that is follows a flat file structure, and it also gives an accession number and gene names. And there is an collaboration with other databases of DDBJ and EMBL and GenBank, which is known as largest collection of nucleotide sequences. Okay, here is from quiz. After this quiz, we'll move on to the EMBOSS demo. So how many different types of nucleotides are found in RNA? Yeah. Kindly answer for A now, later. Yeah, we have time, so we'll take it slow. Yeah, it's correct. AUGC. We have 4 nucleotides in RNA. How many different types of proteins are found? Amino acids are found in proteins. Twenty. How many nucleotides are needed to code for a single amino acid? Yeah, everyone can provide their answers as these quiz are not graded, just for your practice purpose. Yeah, so we have combination of 3 nucleotides coding for a one amino acid, that is codons. how many different codons are there in a genetic code? 64. So how it is 64? Yeah, 4 into 4 into 4. Yeah. So how many codons code for amino acids? 64 is fine. Do all 64 code for some amino acids? Or is there any change in the number? Okay. I see some of them giving answers 61. So what does other 3 code for? Yeah, the other 3. So we have 61 amino acids, 61 codons coding for 20 amino acids and 3 denoting the stop codons. 61. Okay, now I move on to EMBOSS. So is there any clarification you need in the problem solved before? I'll take that. Then I'll give a demo on EMBOSS. Then I'll give you some task which you can perform using EMBOSS. Yeah, please go ahead, unmute yourself. I can see Pavani.
Yes, sir. Good evening, sir. Sir, before you told that we'll be having 3 different reading frames for a DNA sequence to carry out for the protein translation. So I want to know how during translation it will be confirmed that this reading frame will be determined for its translation.
Yeah, actually, a little correction I will make to your question. Each strand of a DNA will have 3 reading frames. So overall, 2 strands of DNA will be having 6 reading frames. Okay.
Yes.
Yeah, and there will be some recognition sequences. Like for example, there is cosec sequence and Shine-Delgarno sequence. So if this sequence is present in the upstream of the start codon, then that is coded as a protein. Like that reading frame is considered.
Sir, does AUG has anything to do with the determining reading frame?
Yeah, like when we observe in a smaller scale genes or genomes, the chance of seeing multiple AUGs before a stop codon is less. But when you go to a complex genomes, you can see AUG followed after 100 sequences, one more AUG. So we can encounter multiple AUGs before the stop codon. So in that case, this Shine-Delgarno or cosec sequences are used as recognitions and reading frames downstream, but exact downstream to that. The next downstream to that is considered for protein synthesis.
Okay, sir, got it. Thank you.
Okay, before going, how many of you are not familiar with these biological concepts? Like the terms like alignment, complementarity. Okay, some most. Okay, please, for those who have raised the hands, please closely follow the demo, what is happening. I'll be explaining right from the sequence to the answer, everything, input to the answer. So this may help you in understanding those concepts. Okay, first I'll start with the EMBOSS online. So you can, everyone can see my Google. So in the Google search bar, you can type EMBOSS Google. The first link you see is EMBOSS program, EMBL and EBI. Just go to that. So initially you see multiple sequence alignment and pairwise sequence alignment. So these sequence alignments will come in next upcoming classes. So we'll see the sequence translation concept. So we type EMBOSS, go to the EMBOSS which is hosted by EBI. European Bioinformatics Institute. So under that we go to sequence translation. Under sequence translation, the first one we see is transseq, which I am highlighting it now. Transseq. Sorry. Transseq. So this translate nucleotide sequences to the corresponding peptide sequences. Further, we have 6-pack. So which displays DNA sequences with 6 reading frame translations and overlaps. Overlaps are nothing but open reading frames. Next, we have back transseq, which back translate protein sequences to nucleotide sequences. Okay, first let me start with transseq. So you can enter the screen is visible, right? Do I need to zoom more for clear visibility? So the step 1 is enter your input sequence. So then in general we input using the FASTA format. So the FASTA format is nothing but header followed by the sequence. So for now I am using an example sequence given by EBI. So you see the first is a header. What I am highlighting here is a header. Followed by that we have nucleotide sequence. And if not, for large files which we can't type or enter, we eBay also provided to upload a FASTA file here. Even you can give sequence input or file as input. Then we just go the frame selection. So just like we have answered the frames. So here the options are given is 1 frame, 2 frame, 3, F, which is forward 3 frames, -1, -2, -3, R, reverse 3 frames, and all 6 frames. So we can select according to our need and according to our study. We can select what frame we require. Okay, for now I'm going with only 1 reading frame forward. Then submit. So it will take some time to give out the result. You can see this is the protein sequence. We have given a nucleotide sequence as input, and now protein sequence. This is one first reading frame. Similarly, other reading frames can also be extracted by selecting the option. Okay, let's see all the 3 forward reading frames. Yeah, so as I mentioned, I'll just give a recap of this. So we go to EMBOSS online, then we select what are the tool we require. Then under the tool, the first option is to input our sequence as FASTA format. Either we can give a text as input or file as an input. See now, previously we have only 1 reading frame. Since we have selected 3 forward reading frames, here we have 3 forward reading frames. And since this is an online version, this has very limited tools or packages available for understanding. So they have a downloadable version of them which has many packages along with the documentation available for use. Okay, I hope this online demo is fine. So if any doubt in online demo, please ask me now. Later I'll move to how to download and use EMBOSS. So now we have the online version here. Similarly, I'll type in the Google emboss again. So instead of going to the first emboss, I'll type emboss download. So here the second option, open Bioinformatics Foundation. So we can click that. Here different about web server and FTP. FTP is nothing but a file transfer protocol. This is a secure way of transferring files. Now it is actually hosted in their server, and this is FTP is a secure way of transferring. So here we have a FTP link available. So here you can see the FTP link available in the top. So one can copy this link address, then move on to their web server, move on to their this PC or file manager. In the search, in the heading where I'm typing on the top heading, you can copy paste this emboss, the FTP link, and click enter and wait for some time. You will be redirected to the emboss website. The first step is copy the FTP link of emboss, then go to your this PC or file manager, whatever in Mac, Linux, wherever, go to your file manager. In the address box, which is on the top box, paste your FTP link. Remove everything and paste your FTP link. Then you will be redirected to the emboss website. So in the emboss website, you can see there is so many new updates. So under Windows section, we can find executable file, and old executables are found in old directory. This is the recent updated one, emboss 6.5. So you can copy this and save in your local system. Actually this is now not in local system, so copy. Okay, I'm saving it in desktop. Then install this executable file. Yeah, while installing, if you feel any error or any error screen is coming, so that is because of Java is not installed in your system. So before installing emboss, kindly install Java, then use emboss. Please be muted. Okay, so I have installed emboss. I don't know how big is my screen visible. So on the left side, you can see some options provided. So under these options, you can see the fifth option as documentation. Yeah, please, someone raise their hands.
Sir, kindly zoom that option, sir. I can't able to visible.
Yeah, actually the zoom is not available in this emboss since it's Java based.
Okay, okay, sir.
Yeah, like I'm just giving the number. The fifth option, we have a documentation. So if we click on documentation, we can see different functions along with their, even we have edit option. So under edit, the sixth one, we can see what is the name and what is the purpose we can use. Below that, we have a scroll bar given where I'm scrolling. So under this, we have so many options given. So for example, previously we have tried transseek, right? Even transseek is given here. So this is transseek. Similar to the online version, what we have seen, we can either paste or upload a file. Then to change the other options. So previously we have viewed options like reading frame selection and all. So the extra additional options are given under advanced options tab. Okay, I'll just demo this. So I have opened transseek from the left side scroll window. So transseek is actually used to translate nucleic acid sequences to protein. So I am uploading a file. So this file I can upload from, so I have a demo file. So I am selecting a demo FASTA file. So you can get what is a demo FASTA file. So here I am showing you my demo FASTA file. This is an insulin sequence I took. It's not complete insulin sequence, it's a truncated insulin sequence only. So this is what present in the gene.fna file, gene.fasta file. So FASTA file has information header on top, so which starts with this symbol followed by the information about the sequence and then sequence followed by that next line sequence. This is a FASTA file format. Now I have uploaded the file. Then I am selecting again a forward frame here, then clicking on go. So here you can see a saved results window appeared. This I can zoom in. So this is the first reading frame of the sequence that we have input. Similarly, we can also get input for 3 reading frames. Yeah, this is how. So in online version, we have limited access to these features. In an installed version, we have n number of tools to use for different purposes. So I see 2 people hand raise. Can I know? Yes.
Hello, sir.
Yes.
Sir, actually, I'm using a Mac PC. So in it, when I clicked on the link, it navigated to the Finder option, and from it is now it is showing all the embossed tools. Means there is tar GZ files are here present.
Yeah, actually the FTP is like you need not click, you should open it through your file manager in Mac.
Okay,
Just like
fine.
you search for your file, right? Okay, I'm searching for this file, like that. FTP is not a direct accessible link, you should access it through your file manager. Even it is Linux, Mac or Windows.
So after direct searching, it will give the folders.
Yeah, it will take you to where you will get a DMG file, .dmg file, which is an installable. I think so. It should give you a DMG file, or it will give a
Okay, sir.
jar file which you can copy paste on your local system to use it continuously.
So I will try once.
Yeah.
Thank you, sir.
Next, I see one more hand raise from. Okay. So this is the brief demo of EMBOSS. So you can explore EMBOSS. So here I have given some task, so which can be done using the EMBOSS. If you can install, and you can take it as home and perform. And if you have any doubts in performing these, okay, you can come back to me in the next class. So now this is what I have planned in this session. Sorry for the little interruption in between. Yeah, so any other questions I can take now. Yeah, so in each of my session, the last 20 to 30 minutes will be demonstration of any bioinformatics tool which is related to the course. Mostly I'll try to follow the week. So with the tools which are related to that week, yeah, I'll demonstrate the tools. Yes, because I understand the same lectures were taken by 3 other scholars as well. So this is what I would like to do extra when compared with them. One more question I would like to ask. How many of you have completed the assignment of week 1, like submitted? Okay. Yeah, Abhilash, I'll repeat. Yeah, Abhilash, you want me to repeat? Yeah, thank you for your answers. I hope many of you have submitted the assignment. No, assignment, this is what the NPTEL assignment. Yeah, you can find in the Swayam portal, the assignment is already active. Yeah. Yeah, Abhilash, you asked me, could you repeat the steps? So what steps you want me to repeat? Steps for?
So just once going from the FTP link.
Okay, I'll show it again. So when you have the FTP link, you just go to your this PC. You can see, this is my just this PC or file manager in your system. Then I go to this above search bar. Then I'll copy paste the FTP link here. Then click on enter. You will be redirected to the EMBOSS page. So here you can download whatever version you need, and there is specific tools as standalones also available for different purposes.